Viral transcripts show diverse isoform in hybrid ecDNA and hybrid transcripts expression. SD226SA409, a site for the production of heterogeneous nuclear ribonucleoprotein, which is a requirement for the production of HPV-E7 mRNA and also an indicator of increased risk of cervical cancer. Note that SD226SA409 represents a junction which creates a shortened form of E6 called E6*1 that will result in a premature stop codon. Human ecDNA tumors had reduced E6*1 production compared to non-human ecDNA tumors, while hybrid ecDNA tumors did not have reduced E6*1 production. Figure 2a and 2b shows the ecDNA structure for individual tumor samples and orders by chromosomal location of the gene status in hybrid ecDNA oncogene, human ecDNA oncogene, no established oncogene, and fold change in expression of the ecDNA plus tumor gene. It is seen that ecDNA associated with human genes from the institutional cohort exhibited upregulation of associated genes in both figure 2a and 2b. The only difference is the start upregulation in 2b compared to 2a. Figure 2c shows the increased expression in association with both hybrid genomes and hybrid transcriptomes. It is determined that hybrid RNA tumors showed higher upregulation of associated genes than hybrid DNA tumors in both institutional and TCGA cohorts. Figure 2D shows the dramatic increase of D41 in TTC33 expression downstream of the HPV human hybrid sequence junction. Figure 3A shows the principal component analysis of splice junction expression in individual tumors with annotation of tumors by presence or absence of human viral hybrid transcripts. It is determined that hybrid transcriptome tumors exhibit distinct splicing patterns compared with non-hybrid transcriptome tumors. Figure 3b shows the genome map of HPV16, DNA, and RNA. It is determined that the E4 to E5 region, including the 5' end of L2, was far less likely to have selective enrichment for genomic copy number in hybrid RNA tumors. Also, there was a decrease in expression of those regions as compared with tumors without hybrid RNA. Figure 3c shows the splice acceptor cluster quantification of hybrid transcripts for institutional and TCGA cohorts stratified based on splice donor location. It is determined that median splice acceptor range varied from 29 to 20,399 nucleotides wide across the TCGA institutional cohort and were narrowest for SD1302 and widest for SD226. Although there was no preferential splicing to SD880 to a human acceptor to be associated with ecDNA status, tumors with either form of ecDNA were significantly more likely to splice to a human acceptor from SD880. Splicing patterns of SD880 in TCGA are analyzed, and it is found that 10 out of 28 are preferentially spliced to a human splice acceptor rather than canonical SA335A. The authors noted a strong association of splicing patterns depending on hybrid DNA or RNA status in the institutional cohort, irrespective of ecDNA, as shown in Figure 1a and 1.